Welcome and thank you so much. This is our 25th anniversary of our Sea Secrets, as you can see, and tonight is our second lecture. I'd like to start off with thanking our sponsors. Bank of America, thank you so much for your support. The Shepherd Broad Foundation, the Meredith Ann Dasberg Foundation, William J. Galway, Cheryl Gold, the Key Biscayne Community Foundation, Elizabeth C. Lambertson Foundation, Joan McCon Family Foundation, Taylor and Melissa White Fund, Myron and Nicole Wang, the Welch Family Foundation, Southern Glazers, and Wine and Spirits. Thank you all for our sponsors that have supported this event. We really appreciate it. And thanks to other supporters too who support the Rosenstiel School globally. And I just wanted to say we really appreciate all the support that you give to us for not only this event but for the things that we do here at the Rosenstiel School. I'd like to begin this lecture by introducing Maria Cartolano. She is from Allentown, Pennsylvania, and she received her undergraduate degree from the University of Miami class of 2013. She then went on and received her Ph.D. at the Rosenstiel School, and she graduated in 2018. Currently, Maria is a postdoctoral associate in Danielle McDonald's lab. Please join me in welcoming Maria.
Thank you. So I started at the University of Miami in 2009 as an undergraduate student. And I had a pretty broad interest in marine science. So I came to this school because I had a really excellent course list of undergraduate courses that I could take in marine science, as well as a really diverse research program at Rasmus. I was able to take a wide variety of classes as an undergraduate, which really helped me find what I was passionate about. So during my junior year, I took a Comparative Physiology course with Dr. Danielle McDonald, and during the first class, she advertised that she had a position open for an undergraduate researcher in her lab. I immediately applied, met with her, and she offered me the position. And I can remember that the very first day that I was working in her lab, she had me doing fish surgeries and starting on my own project. And I can remember that she took the time to explain new techniques to me and new concepts. And she was really encouraging and also promoted independence of my own research. So it was this experience in the lab working with her, as well as the physiology courses that I took with her, that made me realize that I really enjoyed working in a laboratory setting, as well as I was fascinated by physiology. So I graduated in 2013 with a Bachelor of Science in Marine and Atmospheric Science. And that year, Danielle McDonald was also looking for a Ph.D. student. And because I loved working on what I was working on, and because I knew how rare it would be to find a mentor like her, I was so excited to start my PhD with her in 2013. My PhD research was on chemical communication in fish. So toadfish, which you're going to hear a lot about today, have a unique mechanism of pulsing urea across their gills. And this mechanism is unique because most fish just continuously excrete ammonia across their gills as their nitrogenous waste. But toadfish are unique because they pulse urea a couple of times a day, and it has remained a research question for a couple of decades of why they do this. So the goal of my PhD was to provide evidence that they were using it for chemical communication. So I did a couple behavior and physiology experiments to provide evidence that they are using pulsatile urea excretion to release pulses of urea across their gill that might contain chemical signals or pheromones to communicate their reproductive status, meaning that they can communicate which sex they are or if they're ready to reproduce. It might also allow them to attract mates, as well as communicate social status, so if they're a dominant or a subordinate fish. Now, in addition to understanding why they did it, I was interested in how they did it. So I looked at the physiology of this process, and it's controlled by the stress hormone cortisol, which you will also hear Dr. McDonald talk about in a little while. So I learned a lot about stress in fish, and this led me to staying on as a postdoctoral research associate in Danielle's lab studying stress physiology. So I'm now working on a grant where she's looking at the effects of the Deepwater Horizon oil spill on the vertebrate stress response. And I've also worked on a project looking at noise pollution in fish, which is also very stressful to fish. So I understand a lot about stress physiology, and that's kind of why I'm still around. So I'm so excited that you're going to get to hear Dr. McDonald talk about her work today because she's an exceptional researcher, teacher, and mentor. She has won the Rasmus Outstanding Mentor Award, as well as the Graduate Faculty Mentor of the Year Award. She's a great mentor not only because she brings us really good snacks for lab meetings, but because she creates a fun and productive and positive environment for us to work in. No matter how busy she is, she always finds time to sit down with us, to troubleshoot if we're struggling, or to bounce ideas off of each other, and she's always eager to hear about our progress. She cares very deeply about our personal well-being as well as our professional success. No matter if an experiment is going smoothly or maybe not so smoothly, you can always hear from her that we're doing a great job. And it's this encouragement and positivity that keeps us really excited about the research that we're doing in our lab. Dr. McDonald has also been awarded the Linda Farmer Award for Excellence in Marine Science Teaching. So despite her being really busy with teaching and advising and leading our lab, she still finds time to work in the lab and do her own research. So you can find her in the lab doing research for herself or working alongside her undergraduate and graduate students as well as her postdocs. Now, her passion and enthusiasm for research and education is very contagious, and I'm an example of that. She's a great mentor not only to the people in her lab, but also that she serves an excellent role model for women in science. She promotes and contributes to community outreach, so she does a lot of presentations and hands-on demonstrations to people of all ages in the community to tell people about the research that she's doing and the research at Rasmus, which really demonstrates her ability to be a great mentor beyond this university. I am so thankful to her for being a great mentor over the last 9 years, and I'm so thankful to Rasmus for all the opportunities that I've had thus far in my career.
Thank you so much. I would like to now introduce Danielle McDonald. Come on up. Thank you.
Hi, everyone. Typically, Dean Avasar will introduce the speakers, but he has come down with the flu today. So I'm introducing myself in the spirit of the International Day for Women and Girls in Science. So, hi. So my name's Danielle McDonald. I'm a professor here at Rasmus at the University of Miami in the Department of Marine Biology and Ecology. I've been working here since 2006, and most of my research focuses on this guy right here, which is the Gulf toadfish, Opsanus beta. And Maria told you a little bit about some of the other work we do in the lab about chemical communication. Today we're going to talk about what toadfish can teach us with respect to human health and disease. Before I get started, though, I'd just like to acknowledge the people who were involved in the research I'm going to present. I'm actually not going to present too much data today. I didn't want to overwhelm you guys. But even with not so much data, this is how many people contributed to it. So doing science is a team effort, and you can't get anywhere with the help of a whole bunch of people. So I just want to thank them for their contribution to this work. And also, just note that today is International Day of Women and Girls in Science. I've had the pleasure and the luck to work with a bunch of really great female scientists. This is just a few that I've worked with over the past year or so, and I've had lots of women come through my lab, and it's always been a real pleasure. So yay, girls! Girl power! All right, I'm actually going to step down because there's some videos I need to play, and I need to be down here for this, but I'll come back up in a bit. So today I'm going to talk about three main things. I'll tell you a little bit about the Gulf toadfish, some fun facts. I'm going to tell you about the power of comparative physiology and what that is, and then a little bit about using toadfish as a model for human health and disease. So first, the Gulf toadfish. So essentially, the toadfish is just your neighborhood, bottom-dwelling, noise-making marine fish. It's found along the coast of Florida within the Gulf of Mexico, and it sits on the bottom And it makes noises. So if you've been scuba diving, you've probably heard them. There's a toadfish here underneath this cinder block, and it's calling out to females. The females will come to the nest and lay eggs, and the male will stay there and take care of the eggs until they hatch. There he is right there. Oh, he's so nice. Okay, so they produce a lot of sound. They have a swim bladder that they use to produce noise. And you can see them all over the place around here. Like most of you, toadfish are not fans of the Ultra Music Festival. So I don't know if you heard about this, or I'm sure a lot of you lived through it, but the Ultra Music Festival was on Virginia Key last year. At Rasmus, a whole group of us did an experiment trying to see whether the music would stress the fish out. So this is basically what they were hearing in their tanks. Hang on a sec, I'm going to turn this way up so you can hear it. We had tanks outside in the University of Miami Experimental Hatchery just across the street with toadfish in them. You can hear the music. So it sounds lower because this is the sound that was coming through the water into their tanks. But it's still substantial because what they're used to is silence. So this was pretty major. So what we did was, now the bass is coming in. Ooh, I started it again. Like you guys are fans of the Ultra Music Festival. Okay. So what we did was measure levels of stress hormone. So the stress hormone in toadfish is called cortisol. And we measured blood cortisol levels twice before Ultra. So this was 3 weeks before Ultra, a few days before Ultra, and then during the Ultra Music Festival. And you can see that the toadfish experienced a significant increase in stress hormone levels. So they definitely do not like Ultra. And there was a lot of press associated with this work. People got very excited about it. And eventually it led to Ultra being ousted. Well, I mean, the toadfish didn't oust. Ultra decided to leave. They volunteered to leave, and now they're back at Bayfront Park. For most of this, it was pretty accurate reporting, but there was one news report that said the fish were less stressed than being in a crowded tank, but more stressed than if they heard the sounds from toadfish eating dolphins. I mean, toadfish are this big. Dolphins are this big. So this wasn't happening. But toadfish are actually pretty vicious. So you don't want to be on a toadfish's bad side. They wouldn't be eating dolphin, but they're seriously pretty mean fish. I'll show you a little video here. I'm just going to pop it up a bit. And this is 2 toadfish here in the back interacting. We do these behavioral experiments where we kind of force them to interact. They have to fight over this shelter, and there's a dominant and subordinate fish. And you can see that they're pretty vicious. He wants to get out of there. They're not too bright. They're vicious, but they're not too bright. Okay, so if Maria was doing a behavioral experiment, for example, she would be scoring these 2 fish and giving them points for aggressive acts or negative points for subordinate acts. So this guy is the aggressor. He's the one, he moved towards the back, and that one that kind of fell on him trying to get away is the subordinate fish. Anyway, you don't want to be on a toadfish's bad side. They bite. That's my finger. And this is the guy who did it. He was very angry. And I do think they learned this behavior very early on. These are all toadfish. They're all alive. This is the daddy toadfish with a bunch of babies. And look at them. They're all ready to take your finger and give it a good bite. So they're learning this behavior very early on. But toadfish do make a good model for the study of human health and disease, and they have a lot of really interesting attributes that make them a good comparison to what may be going on with human health. A very close relative of the Gulf toadfish, the oyster toadfish, was actually taken up in the space shuttle to look at its vestibular system, which is what's involved in balance, because their systems are very similar to ours. As I've shown you already, they make sounds with their swim bladder. Their swim bladder muscle is the fastest twitching muscle in the animal kingdom. They have a really cool kidney. It's very specialized for living in a marine environment, but it makes it a lot simpler than our kidneys, so it's very good to use as a comparison to what's going on in our kidneys. They make urea as their nitrogenous waste like we do, and that's part of liver function. So they have a liver that's more similar to us than other fish. They have red blood cells that sickle, so they could be a very good model for sickle cell anemia. They have the neurotransmitter serotonin in their bodies, like most other fish, and we're studying how serotonin is regulated, and so serotonin is a very important neurotransmitter with respect to depression. They get very stressed out, as you saw with respect to ultra, and we use them as a model to study stress in marine mammals. And they're very tolerant to ammonia, which is a nitrogen waste, and they can make a very good model for something called hepaticencephalopathy, which is your brain swelling due to liver failure. So for all these reasons, we like to study them. But you might be sitting there saying, why are we talking about human health when we're at a marine school? Isn't there a med campus just down the street from us, right? Shouldn't we be focusing on this kind of stuff? And that leads me to part 2 of my talk, which is the power of comparative physiology. So comparative physiology is, I'm going to go back up here. I hate being by a podium because it's so constricting. Can you guys still hear me? Yes. All right. So comparative physiology is essentially learning more about how living organisms and their parts, so their organs, function by comparing their similarities and differences. So, for example, okay, so in this way, if you take comparative physiology into account, marine organisms can sometimes make the best model to study human health and disease. So here we have a toadfish, your typical white laboratory rat, and my favorite postdoc, Maria Cardolano, who I was always going to say that even before her wonderful speech. All right. So sometimes marine animals can make the best model because they're more similar to a human than another mammal. So take stress physiology, for example. I just showed you how toadfish, they have the stress hormone cortisol. Humans have the stress hormone cortisol. But your typical white laboratory rat has something called corticosterone. So here we have a fish that's more similar to a human. Now, you can use comparative physiology in a different way. Sometimes marine animals might make the best model because they're completely different from a human, and you can learn a lot about how the human works by studying the differences of the fish. So in the case of toadfish, toadfish are super tolerant to ammonia. Ammonia is what builds up if you're in liver failure, and it causes your brains to swell. But both rats and humans are intolerant to ammonia. So we use kind of the concepts of comparative physiology to figure out why toadfish are superstars with respect to ammonia tolerance, and how can we learn from their tolerance. So comparative physiology, or the ideas of comparative physiology, were really first established by a French scientist called Claude Bernard. He was, I think, born in the 1820s, so quite a while ago. And he stated that in scientific research, the tiniest processes are of greatest importance. The lucky choice of animal, of an instrument built in a particular way, the use of one reagent instead of another often suffice to solve general questions of the highest order. So he proposed that the type of animal matters when you're asking and trying to answer scientific questions. This was followed up by another famous scientist, August Krogh, about 50 years later. He was a Danish scientist, and basically he stated for many problems, there is an animal on which it can be most conveniently studied. And this idea is called the August Krogh principle, and it's a very important concept in physiology. Basically, you can find an animal that's going to work for your question. August Krogh actually took this to another level. So this is a picture of him with a grasshopper carousel. So he built like an entire airtight room, and he attached grasshoppers onto this carousel to measure oxygen consumption in insects. He was famous for doing all sorts of work on all kinds of different organisms and building things to be able to test what he wanted. Eventually, he won the Nobel Prize in 1920, I think, for physiology and medicine by using frog skin and establishing what capillaries were for. All right? So many Nobel Prize winners have used animal models to study human health and disease, and many have also used marine animal models for the study of human health and disease. So some examples are starfish larvae. So this was a model used by someone called Mechanoff, I think in 1908. And he discovered phagocytosis, which is essentially how an organism can engulf something and bring it into a cell. So this is a very important concept for the immune system. That's how kind of our leukocytes grab a bacteria or something and bring it into the cell to protect our bodies. So he discovered that by using starfish larvae, which are transparent, and it was very easy for him to tell that. Another famous example is the ionic basis of nerve conduction, which was discovered using the squid. So this was done by a duo called Hodgkin and Huxley in the 1960s. They also won the Nobel Prize. And essentially, they used squid because squid have giant nerve cells. So here's the start of the nerve cell, and they propagate all the way down here. It's not the squid that's giant, it's the nerve cell that's giant. Our cells are also very long. We have nerve cells that can be up to 1 meter long, but they're very, very thin. Whereas squid giant axons, you can see it here in a dish, it's about 1 millimeter across. This was massive compared to human nerve cells. And it enabled us to learn about how nerve impulses were conducted across a cell. And it requires the movement of sodium and potassium and the opening and closing of channels. They were able to determine all that, and they established what is known now as the action potential, so this change in voltage that occurs across a nerve cell. This again was done using squid, and it could not have been done using any other organism at that time. Another great comparative physiology example is the sea slug. So we have Aplysia actually here at the University of Miami. Eric Kandel used the sea slug, Aplysia californica, to determine the cellular basis for learning and short-term memory. And the reason he used sea slugs, I actually love saying that word. I don't know why. Okay, the reason he used sea slugs was because human brains have 10 to the 12 neurons. So over 1 billion neurons. An Aplysia brain have 2 times 10 to the 4th neurons. So much simpler brains. And what you can do with Aplysia is look at the very cellular level what's going on in terms of movement and what's going on in terms of memory at the cellular level. How do you learn? How do you remember things? How does the cell change? And you can only do that in organisms that are much simpler because there's just too many cells to look at in the human brain. All right, so that brings me to my own research, which is looking at toadfish as a model for human health and disease. So today I'm going to talk to you about 3 projects that we've been doing in the lab. The first is looking at serotonin handling. The second, ammonia tolerance. And the third, cortisol secretion. And we're actually, for cortisol secretion, we're using toadfish as a model for bottlenose dolphin, which is almost like a human. And it was in my little blurb when I wrote it up for this talk, so I've decided just to include it, even though it's technically not human health. So serotonin. Serotonin is a neurotransmitter most commonly known for its role in depression. And so let me tell you a little bit about how that works. So right here we have 2 nerve cells. So these are similar to what I was talking to you about with respect to Hodgkin and Huxley and the squid giant axon. So same thing, this is a nerve cell, and this long part is actually the axon of the nerve cell. And 2 nerve cells will communicate with neurotransmitters. So an electrical signal will move down this axon, and it will cause the release of neurotransmitters from one cell, and those neurotransmitters will move and bind to this second cell, and the nerve signal will propagate forward. Let's just look at this area right here. So this is nerve cell number 1, and this is nerve cell number 2. And essentially, these little circles are containing the neurotransmitter, and that neurotransmitter gets released into this space here between the cells, and it's called a synapse. So if this is serotonin, and there's all kinds of neurotransmitters, but I'm obsessed with serotonin, so we'll talk about serotonin. Serotonin gets released in the synapse, and then it binds to receptors on nerve cell number 2, and nerve cell number 2 will then have an action potential. When you are depressed, you're often prescribed something called a selective serotonin reuptake inhibitor, or an antidepressant, such as Prozac, is kind of your tried and true antidepressant. Those antidepressants target this transporter right here, whose job is essentially to recycle the serotonin. So the serotonin gets released into the synapse, and then it goes back into the cell and gets repackaged into these vesicles to get released the next time around. SSRIs, or antidepressants, will inhibit that transporter and cause the buildup of serotonin in the synapse, and it tends to make you feel better. It's actually never been completely proven that it is the buildup of serotonin within a synapse that reverses depression. And it's never actually been proven that people who are depressed have reduced levels of serotonin in their synapse. And that's because you can't measure this in humans as they are experiencing depression. Everything we know is based on animal models. And we do know that drugs that tend to inhibit this transporter make people feel better. So we're kind of putting the puzzles together. All right. So that's serotonin and depression, but serotonin actually does a whole bunch of things in our body outside of the central nervous system. So serotonin and serotonin transporters, so that protein that is taking serotonin back up into the cell, are also important outside of the brain. This is just a blood sample. And you can see, can you guys all see these little green dots? Yeah? Okay, so these little green dots are actually smaller cells in our blood called platelets, and they're involved in clotting predominantly. But in this sample, what we've done is put the blood, incubated the blood with a dye that will move through serotonin transporters only. And so what this means is that these platelets have serotonin transporter in them, and they're accumulating a whole bunch of dye. And in fact, if you measure platelet serotonin concentrations, they have very high serotonin concentrations. It's believed that serotonin in the platelets is involved in blood clotting, because that's what platelets do, but it's also involved in controlling blood pressure. Because if you had serotonin just floating around in your blood, it causes massive constriction of your blood vessels and high blood pressure. So this is a very important safety mechanism that we have. This is data from fish, but you would expect the same thing from mammals and humans, where if you inject fish with an antidepressant, so you block serotonin transporters, you see a very big increase in blood pressure. Okay, so you prevent serotonin transporters from doing their job, you get a big increase in blood pressure. They need to be working in the platelets to sequester the serotonin to control blood pressure for us. In fish, there's no platelets. Instead, they have very high serotonin levels in their heart. So serotonin is staining here green. And they have high serotonin levels in specialized cells in their gill. So here we have fish acting very differently than humans. And they're using cardiovascular tissues, their gill, which is involved in oxygen exchange, and their heart, which is involved in pumping blood, to control serotonin levels. We also find very high serotonin transporter levels in the heart, up to 30 times more than the brain in fish. So what we're really kind of interested in doing, I'm in particular, to be honest, I'm very interested in what serotonin is doing in fish, period, because people don't know what it's doing. But in terms of a human health model, there is a link between depression and cardiovascular disease. And so here we have a potential model for human health where we have lots of serotonin transporter in cardiovascular tissues. What is it doing and what can it tell us about a system that's completely different, which is the human? Another element of this work of looking at antidepressant effects in fish is the impacts of environmental pharmaceutical contamination. So this is wastewater entering into a waterway, and everything that we use ends up in wastewater. And it's now found in measurable quantities in coastal waters, in rivers, lakes, even in drinking water. So this is my kitchen counter, actually. No, I'm just joking. But you know what they say, Americans can be separated into two people, those that are medicated and those that should be. So we're having more consumption of, as mental health is being on the radar more and more, people are getting out there and they're taking these compounds, and that is a good thing. But it also means that more and more of these compounds are being found in wastewater, and that is a bad thing for fish. But what fish can be used for is a model for long-term pharmaceutical exposure, because they are in low concentrations of pharmaceutical over the course of their life. How do things change in them if they're constantly being inundated with pharmaceutical exposure? So that's another thing that we're doing in my lab. All right, the second component of research that I do that involves human health is looking at ammonia tolerance to avoid hepatic encephalopathy. And this was actually started by Pat Walsh, who is actually here today, who is my postdoctoral supervisor. And people in this field say this common saying, when the liver fails, the brain ails. So now you guys can go up to liver scientists and say that and sound very cool. Essentially what happens is when the liver fails, either due to cirrhosis of the liver or due to inborn genetic errors or any other issue, gunshot wound, it happens in TV shows, you end up with high levels of plasma ammonia, and that is toxic to your brain. If you are in liver failure, the cure is to get a new liver. So this is a very important disease. So this is a normal individual, and this is somebody who's suffering from hepatic encephalopathy. And you can see, if you look in this area right here compared to here, how much more white there is here. So that's brain swelling. This eventually causes seizure, coma, and death. What we had found several years ago is that toadfish brains do not swell in response to high plasma ammonia levels. So we put toadfish, this was with Pat Walsh in Belgium, we put toadfish in these mini MRIs, magnetic resonance imaging machines, that were actually made for songbirds. And so these guys, these Belgian researchers, were not thrilled that we were putting toadfish in there and running seawater through their very expensive machine, but they took it. And we found that toadfish brains do not swell in response to high ammonia. So this is scans that we did. We had the MRI experts read the scans, showing no swelling. And this is essentially what a toadfish brain looks like versus a human brain. So human brains, there's a big kind of diencephalon here. And a toadfish brain, our brains are like this, and toadfish brains are kind of flattened out. But they have all the same parts as we do. So what I've been doing in the lab is trying to grow specific brain cells from toadfish to study them further. It's cells called astrocytes that swell in humans, and so I'm isolating those cells in toadfish and trying to figure out what makes them not swell. So essentially we use sterile conditions and plates, and in these plates there's a solution that's very similar to blood, but it is not. And on the bottom of these plates, you can get the growth of cells. So these are toadfish astrocytes under the microscope. And these pictures are just showing what rat astrocytes look like, and they're being stained for this protein called GFAP, which is very indicative for astrocytes. And these are my cells, and you can see that they're also lighting up for GFAP, showing that I do have astrocytes. Now, this work is still very preliminary, but what we're going to do is determine whether these cells swell or not, and figure out what makes them not swell. And then ultimately, through gene editing techniques or through pharmacology, maybe we can make mammal cells into toadfish cells and make them not swell as well. And then the last area of research I just wanted to touch on is using toadfish as a model for dolphins, for bottlenose dolphins. And what we've been doing is looking at Deepwater Horizon oil and how exposure impacts the stress response. So this work was was inspired by a study from Schwacke et al. In 2014. And they looked at two different populations of bottlenose dolphin. One population that was exposed to oil during the Deepwater Horizon oil spill, and one population that was not. And they took blood samples from these dolphins. And to do that, you kind of had to circle them with a bunch of boats, and you have a whole bunch of divers in the water, and you round them up. And it's not a very happy experience for the dolphins, so they get stressed out. And they also have the stress hormone cortisol. What Schwacke et al. Found was that cortisol levels in dolphins that were not exposed to oil were elevated as they should be, undergoing this very stressful experience. But dolphins that were exposed to oil had much lower cortisol levels. So they were not getting stressed out the way they should be. And you may think that stress is a bad thing, so it'd be great not to get stressed out. But in fact, Stress is what helps us survive. We all need to be stressed at some point, to get ready for a test, to get ready for a talk. You need stress to get you through what you have to get through. And for these guys who are not getting stressed, that means that they are not turning on everything they need to survive the oil exposure, and that is a problem. So what we've been doing in the lab is using Toadfish as a model to study what is going wrong with the dolphins, because people don't like you putting dolphins in labs and taking blood samples and stuff. So we use Toadfish instead. They take up a lot less room. And we do a lot of sampling, and we take blood samples from Toadfish. We take a whole bunch of samples, and we are measuring a whole bunch of things. But essentially, the bottom line is that after 7 days of exposure in Deepwater Horizon oil, you see a significant decrease in the ability to get stressed. And that does not return even after being in oil or being in clean water for 7 days. So they're unable to recover. And this is something that has been found with dolphins too. They're having a lot of trouble recovering from that oil spill. So this is a permanent thing. It may be permanent. Now, the stress response is super complicated, and I'm not going to go into details because it would bore you to death. But it takes multiple regions of the brain. It takes special cells in the kidney, and then within those cells, there's a bunch of different processes that have to happen for cortisol to be released. What we're doing in the lab is trying to look at every single one of those things, and we're down to one last receptor where we hypothesized that it was going to be that receptor, and it's probably that receptor, and we're just trying to figure out the right experiment to test it. So that's basically it. I've told you a bit about the Gulf toadfish. I've told you a bit about the power of comparative physiology and how we use toadfish as a model for our human health and disease. So I hope you're able to take away just little bits and pieces of all those areas. And just one last thing is that the ocean may be full of yet unrealized models for human health. So it's very important to protect our oceans because we don't know what's out there. There could be the perfect model for whatever disease. So thank you very much, and I'm happy to take any questions.
Thank you, Dr. McDonald. We'll take any questions you have. If you do have a question, please raise your hand and we'll come around with the mic for you.
Thank you. Right over there first.
How long do the toadfish live?
Where are you? Oh. How long do they live? That's a really good question, actually. I don't know. Oh, do you know? 10 years? Yeah, I was going to say a decade. Dr. Pat Walsh is the toadfish expert. Yeah, about 10 years. They get to be about this big, and it takes them 2 years, and then their biggest is about this big. So they're not too big. Although I wouldn't want to run into that big of a toadfish because it would bite you and it would really hurt. Oh. I'll let them in.
Mine's a simple question. Where do you get the toadfish?
So it's actually more complicated. We normally get them as bycatch from shrimp fishermen, just locally. Although we have had recently a lot of trouble getting them as bycatch because Biscayne Bay, well, I shouldn't say that. The shrimpers are having problems finding shrimp. And toadfish go where the shrimp go. So for the past 10 months, we had a lot of trouble getting toadfish from shrimp fishermen. But they're back. Good news, they're back. And it just happened recently, actually. But usually as bycatch.
Have you noticed any sex difference in the responses of the toadfish to your manipulations?
That is to the research that I've done here. We actually take sex into account. At this point, I don't think for the Deepwater Horizon work, we haven't seen any differences. Neither for the serotonin work or the ammonia. Maria's work, which was mainly on chemical communication, we were very interested in how males and females responded with respect to that, which is really a whole different story. But I showed you the video of a toadfish calling. Essentially, our hypothesis is that toadfish that are calling out to females are very loud. And so maybe because calling to the female to the nest is so important for reproduction, maybe they'll switch to releasing a chemical to lure the female in if it's not advantageous to be calling. For example, if a bottlenose dolphin is around, which is a predator for them. So in that respect, we might be seeing male and female differences because females do not call to the males. And females are not quite as aggressive as the males are. But we haven't found it in the human health work at this point.
I noticed that in the pictures that toadfish seem to be bottom feeders. And I'm wondering if you find that they're suffering in proximity to humans and the bottom of the water areas where they live. Do you have anything to report on that?
Well, it is something that we think about because when you're talking about pollutants in the environment, a lot of pollutants will end up in the sediment. And so fish that are on the bottom will get exposed not only through the water, but also through the sediment. So we have done Deepwater Horizon oil experiments looking at sediment exposure of the fish. They actually, in our experiments, we didn't see any difference, but there's some caveats to that I could bore you to death with, but I won't. But it is something that we do take into account when we're thinking about pollutant exposure and pharmaceutical exposure, for example. They could be suffering more, is the short answer. Yes.
You mentioned about the inability of the dolphins to recover from the Deepwater Horizon, from the oil, recovering their stress levels.
Right. Where are you? Okay.
And what I was wondering is, have you been able to continue that study onto the offspring? Does it continue into the offspring, that inability? Did they transmit it to the offspring, or it's only for them?
That is a really great question. We have not studied that in toadfish. With respect to the bottlenose dolphin, they are having problems reproducing. So it's hard. That is kind of the end point there, is that reproduction has really slowed down in the populations of bottlenose dolphin that have been exposed. The cortisol and the stress response is very intertwined with the reproduction hormones, and you do need cortisol to stimulate reproductive hormones, and too much stress will inhibit reproductive hormones, but there's a fine balance there. So yeah, that's what's going on there. It's a good question with respect to fish offspring, and I'm not sure if other groups have been looking at that directly. We have not, but there's lots of literature out there. There's been so much research that has been funded because of that spill. I mean, that is, I guess, the good thing that's come out of that, is that we know a lot about oil exposure and the impact. So it's probably out there. I don't know the answer though.
Yes, I have two questions. The first is, what were the characteristics that you measured to determine the stress levels in a toadfish vis-a-vis the Ultra Festival? That's my first question. And my second question is, may I borrow your slide to take to City Hall?
Sure. Well, we were actually like a touring group of talking to city officials at the time, but we were just measuring plasma cortisol. We actually did take a whole bunch of samples, and Maria was doing most of that work with two other researchers, Christina Pasparakis and Rachel Hewer. And they were doing most of the sampling. They had to do it in the middle of the night. And at the time, I mean, pre-Ultra was okay, but during Ultra, I was told that it was pretty awful because the entire room where they were sampling was shaking because of the base, and they were right next door. There was like this second stage on Virginia Key right next to the experimental hatchery. So we really only measured, we have only measured at this time plasma cortisol levels. But we did take we took all kinds of samples just in case, because plasma cortisol, I showed you that picture. It's amazing that we were able to measure an elevation in plasma cortisol in response to Ultra, because you need to have an elevation in all these things to have that happen. So plasma cortisol can sometimes be an unpredictable measurement because there's so much variation between different animals based on this whole complicated axis. So we took all these samples just in case we didn't measure it at the cortisol level so that we can kind of backtrack and see if it was upregulated somewhere else. But we haven't measured those. Yes, you can borrow my slide.
Excellent. We have a question over here, and this will be our last question for the night.
How do you know which dolphins were exposed to the oil spill?
It's a very good question. So they were completely different populations. So one was in Barataria Bay, and that was the oil-exposed group. And one was in Sarasota Bay that did not get any oiling. And dolphin populations stick to particular environments, and they're very well tracked. So they could tell, they know that they stayed where they were supposed to stay.